Yep,
Okay, awesome.
I'm ready.
So Dr. Heather Patisaul is a member of my committee, and she is a prolific researcher with well over 100 publications and an insane H index in the 60s. And what I really am excited about with Heather is that she has done a lot of the research that my dissertation project is based off of. And she's really knowledgeable about endocrine disruption and the way that estrogen receptors impact oxytocin signaling, which I'm particularly interested in. So I'm really excited to have her today. And of course, she also has this big title, and she's a big poobah and all that. But mostly, we're just having her here because we love her. And so, yeah, take it away, Heather.
Well, thank you for that kind introduction. And I want to thank everyone on the call because I missed the last one because we had like the mother of all weather systems come through and like knock everything out. It was total chaos. And now we have a drought, so it's much better for Zooming today than it was last time. So I appreciate your patience and for the opportunity to like try this again and see if we can get it working this time. And so let me share my screen, put it in presenter mode. All right. Sophia, does that look good on your end?
Yes.
All right, perfect. All right, so I want to talk a little bit about how my laboratory and other folks in my division at NIEHS are thinking about how we link environmental exposures to neural developmental disorders. And so we know that the prevalence of neural developmental disorders is going up. So right now, 1 in 6 kids in the US has some form of neural developmental disorder. 1 in 54 have autism spectrum disorder. The numbers are higher in boys than in girls. And every time the CDC checks, that number seems to go up. We have over 350,000 chemical suspects that may be contributing to some of these issues. And there have been multiple studies in humans and animals that have linked things like air pollution, pesticides, and endocrine disrupting chemicals, which are abbreviated EDCs, to autism spectrum disorder, but there really is no smoking gun, right? And so the overall goal of my lab and for a lot of labs in my division is to try to understand how developmental exposures to different kinds of pollution might be contributing to neurodevelopmental disorder. So how do we do that, right? So the first thing is in my lab is we have to recognize that there's a lot of overlap in what constitutes a neural developmental disorder. They have overlapping traits. And so instead of thinking of them as like separate disorders, it's functionally better to be thinking about them as these overlapping traits that then can manifest as a specific disorder, right? So ADHD, the H is hyperactivity, but somebody with dyslexia might also be hyperactive. Somebody with autism may also be hyperactive. And so thinking about these things as overlapping type traits. And the geneticists have really been thinking about this for a while. And so it's well known that there are shared genetic bases for many neural developmental disorders. So there are risk genes that are shared between, like, for example, schizophrenia and major depression. So we have to be thinking about that on the behavioral phenotype side as well. So genes versus environment, this is still an issue. I'm shocked. So even when I came into NIH, there were people at NIH that are like, no, there's no way that any of this can be the environment. Like, that's totally impossible. None of these chemicals cross the placenta, so it's totally fine. And I was like, what? So we know lots of chemicals cross the placenta and get into the developing brain. We also know that chemicals can impact the placenta and alter the development of the fetus. We know that chemicals can impact mom's health and that mom's health may impact the developing fetus. And so the graph that I'm showing here is from one of these hardcore geneticists who's begrudgingly admitted that as much as 40% of the risk factors for a certain disorder, in this case it's autism spectrum disorder, may be coming from the environment. And that number is all over the place. And so what I'm really interested in is that red intersecting area, because honestly, it's going to be an intersection of gene by environment, unless we're talking about a truly genetic disorder. So the umbrella for autism spectrum disorder has been broadened. So it went from a little personal umbrella to like a golf umbrella, and they put more stuff underneath it. So fetal alcohol syndrome is under there now. So that's 100% environmental. That's determined by alcohol consumption. Congenital hypothyroidism, which was used to be called cretinism, So that can be something that's genetic from the mother, or it can be an environmental exposure that contributes to that. Fragile X, right? So that's fully 100% genetic. But most neural developmental disorders are defined in terms of their behavior and do not have defining pathology, which is a real challenge for toxicologists. They're used to looking for pathology. And if you have a aging disorder like Parkinson's or Alzheimer's disease, you're going to have pathology. You're going to have plaques and tangles. You're going to have Lewy bodies. You're going to have something that says this is a sick brain. But if you look at a brain with major depressive disorder, it looks exactly the same as someone who doesn't have it. So that's a challenge for toxicologists. And then these overlapping characteristics that I already referred to, like anxiety and learning disability, and they're often comorbid with non-neural traits. So allergy is a common comorbidity, GI problems are a common comorbidity. They tend to run in families, but are genetically they're idiopathic, right? So even if you do twin studies, yes, there is a higher risk for that identical twin, but it's not clear why they're running in families. It's not necessarily 100% genetic as to what is involved. So there is a role for the environment. The question is just how big is it and what are the chemicals that we're thinking about? And so for the purposes of this conversation, I'm hoping that I can convince you that for brain, that we've all heard that the dose makes the poison, which is kind of true. If it's a poison, a real poison. But for an endocrine disruptor and a lot of different chemicals that alter different points of development, it's less linear than that, and it's more about the timing of exposure. When is the brain sensitive during development? When is the moment in your lifespan where you're most sensitive to these chemicals? It's usually when we're building things within the brain or refining things within the brain. So there may be windows in development where we're not as sensitive and windows of development where we're super, super sensitive. And we could have sensitivities in one sex versus the other. So for me, it's not an either or discussion. The prenatal environment is important. The postnatal environment is important. Genetic factors are important, but also that timing of exposure to environmental factors as well. So if you look at the DSM-5, this is for autism spectrum disorder. That's going to be the example that I use today. You'll see a description that looks kind of like this. So the first thing you'll notice is it's all behavioral. So anyone that is diagnosed with autism spectrum disorder, you're talking about 100% behavioral diagnosis. You may or may not have intellectual disability or global developmental delay. Impaired social communication is a hallmark factor, and so is restricted repetitive behaviors, right? Okay, but every autistic individual looks very, very different. And if you have autistic members of your family or friend group, you know that really well. And so some individuals have highly repetitive behaviors, and for others, it's not a big deal. It might just be a little fidgeting. Some may have major sensory difficulties. Some kids with autism cannot handle really loud noise, or they can't handle really bright lights. Others are fine. Some have anxiety, some don't. Some have language impairments, some have none. So it's very, very heterogeneous. And in your mind, when you're thinking about autism, you're probably thinking about boy autism. Girl autism looks very different. So the behaviors that show up in autistic girls can look very different than they are for boys. They may be hypersocial, whereas boys may be a little bit more withdrawn. And symptoms in girls tend to show up a little later than they do for boys. And that's not surprising. So every single mental health disorder that we have skews towards one sex or the other. So autism, ADHD skew male. We have more males with ADHD and autism than females. In females, we have more eating disorders and multiple sclerosis and some forms of schizophrenia. So it's not terribly surprising that we have a sex skew. And so when you expose with environmental chemicals, you should almost expect the same thing. That also makes sense because the circuitry underlying these behaviors is also highly sexually dimorphic. This is an extreme one, and it's a really reductionist diagram, but I'm just using it for an example. So this is social recognition. So I've met Sophia before. She's met me before. I have good social memory of her. It's a positive social memory of her. She may have a meh social memory of me because I had a bad weather situation and missed this the last time. I don't know. So we're all using the same chemicals and molecules in our body to encode that memory. So we're both using estrogen receptors, we're using androgen receptors and oxytocin receptors. We're using our hippocampus to try to remember and put some valence around that. We're using our medial amygdala. So I know whether to be fearful of her or happy when I see her. But we're both female, so we're not using the lateral septum. If we had a friend who was a male, he would be incorporating the lateral septum in that social memory. And so it's some of the same molecules maybe in both sexes, but different brain regions. So you should always think about sex differences in your experiments. And you should always expect sex differences in the results. And so finally, this article came out in 2024, and I love it. I've been using it in talks ever since because I love a good challenge. And so the public is like, why the heck do you keep using mice? I don't understand it. My kid isn't better, and we don't understand autism more than we did before. And you know what? Honestly, that's a fair criticism. And I think somewhere along the way, as scientists, we've kind of gotten lost a little bit about choosing the right tool to answer the question of interest. And the genetics, I mean, mice are great for looking at genetics. You can manipulate them, you can do all kinds of things with mice, you can make them incredibly fancy. So in some ways, a lot of science just defaults to mice now without putting a lot of thought into, is that really going to be the right tool to answer my question and how translatable is it? And so I think it is fair for us to kind of take a step back and ask ourselves, what am I trying to do? And how can I get there? And is a mouse the right tool to answer my question? So a lot of people will publish a paper that say they have a mouse model of autism or an autistic mouse. If you get me as a reviewer, I'm going to say, no, you don't. Nuh-uh. Uh-uh. So some mouse models can be great. If you're trying to model Fragile X, for example, that's totally genetic. Can make a great mouse model of that. The individuals that have Reelin deficits, another genetic deficit, easily modeled in a mouse, no problem, totally the right model. But some people use they use wild type strains and then use heavy doses of like valproate or methylmercury. That's not how human beings get autism. So it's not going to be a way that humans get autism. Or you might use a funky strain of mouse, like the BTBR mouse is common or the BALB/CJ, because they have autistic traits. Like they might be stereotypic and make a lot of stereotypy in the cage. But they have pathology that you do not see in autistic individuals. So some BTBR mice completely lack a corpus callosum, which is the white matter that attaches the left and the right side of your brain together. They also have deficits in the hippocampus and in the amygdala. So that's not what you would expect in a human. They also lack key human typical social traits like monogamy or bonding or paternal parental care, right? And so is there a better way to go about this than just reaching for the mouse? And if you do use the mouse, you should be aware of some of these limitations and just be honest about them. And so my lab and some other labs have been thinking, is there a better way to kind of approach these questions? And we found the research domain criteria. So this is an initiative that was started by the National Institute of Mental Health. And they started it for kind of similar reasons that we're interested in it, which is what is the biological basis for some of these disorders? What's the biological basis of autism? What's the biological basis of schizophrenia? What's the biological basis of major depression? It's not easy. And so what they did is construct this framework that runs all the way from genes, molecules, and cells up to behavior and self-reports to try to explore the basic dimensions of functioning that span the full range of behavior. So now if you get away from this idea of I'm going to focus on a disorder to focusing on traits, now you can break down the disorder that you're interested in into these specific traits, and then you can model how your chemical impacts those traits. And so you may not be working just on autism, you may be working on autism plus ADHD or whatever, depending on what behavioral trait you're working on. Same if you're in genetics. If you're in a shared genetic variant for multiple diseases, you're working on neurodevelopmental disorders broadly, not just one in particular. So this is what we use. And if you go online, you can dive into these domains. And so I'm going to dive into one that we look at that's relative to autism, which is systems for social processes. And so online, all these blue things are clickable, right? So I'm just sort of illustrating this, but online they're clickable. And so social process, well, what are you talking about when you say social process, right? Like, are you talking about social awareness? Are you talking about social communication? So in our lab, we're interested in affiliation and attachment. So I highlighted that there. And so you can see that this is a framework that'll show you the molecules, cells, circuits, et cetera, that are associated with that particular type of behavior. So if I dive in, here's all the molecules, cells, and circuits. And so me and Sophia's favorite molecules and hormones are in here. So we've got oxytocin in here and vasopressin, dopamine, that sort of thing, different brain regions. But down at the bottom is what's most important. These are the behaviors that define this attachment formation and attachment maintenance. So if I'm going to use an animal model, and I'm going to try to break it with chemical exposures, this is the behaviors I need to focus on. So the first one is preference for an individual, right? I met Sophia, we're hanging out. If I go to a meeting, I'm going to sit next to her, right? Like, oh, I know her. I'm going to go hang out, right? So I'm going to maintain proximity, and then she's going to leave, and I'm going to go, oh, now I'm at this meeting all by myself. Distress upon separation. Same with a partner, same with a sibling, same with your bestie, right? Same with a parent. So mice don't show many of these behaviors. Rats don't show many of these behaviors, but some other animals do. So now I can look for an animal with these three domain criteria. I need an animal that once it learns who another animal is, it'll maintain proximity with a familiar, it'll show a preference for that familiar, and it'll be distressed when that familiar animal goes. And so the species that we work on is the prairie vole. So they're a wild-derived monogamous rodent. So they're up there at the top. And what's really cool about them is there's lots of different species of voles. So they have lots of vole species cousins. And their cousins are not monogamous. So the meadow voles are not monogamous. They behave like rats and mice. They'll come together, they'll mate, the male leaves, female takes care of the offspring on her own, and then she'll mate with a completely different male when she feels like it. But the prairie voles will meet, bond, mate for life, and then both parents, both the mom and the dad, will show parental care. And in the wild, the offspring will also show sibling care. So the first litter will stay with mom and dad. They'll have the second litter. The old offspring will help, and then they'll move on. And so they're way more affiliative than their cousins, the meadow vole, and also rats and mice. So they're monogamous, they're biparental, both sexes mourn the loss of the partner. Sometimes they'll stop eating and die. Some will do okay, but they won't mate again. Others will mate again. It's kind of variable, just like it would be in humans and any other semi-monogamous species. So now, with my prairie vole, I have all three traits. They maintain proximity, they show partner preference, and they show distress upon separation. Even better, the neuroendocrine circuitry has all been worked out. So this is some classic autoradiographies from Larry Young's lab in the late 1990s, showing the difference in density of both oxytocin and vasopressin receptors in key areas that control these behaviors. So now I understand the behavior, I understand the neuroanatomy that's underneath it, and I know what I have to look at. And even more exciting is this neuroendocrinology is similar to primates. Someone else has worked this up and looked at not just human brain, but other monogamous ape brains and said it's exactly the same. So I have a translational species now that I can work on with this question. So that's what we do. We work with prairie voles. This was the species that was instrumental for developing the oxytocin nasal therapy for autism spectrum disorder, which works for a small subset of individuals. So the chemical that I'm interested is a mixture called Firemaster 550. So this was a mixture that was introduced in the early 2000s to replace the PBDEs that were being phased out because they're thyroid disrupting. So Firemaster 550 is a mixture. Most flame retardants are a mixture. So about half of the mixture is organophosphate flame retardants, which is the top two that you can see there. So there's organophosphate esters. You can see the different structure to the brominated flame retardants, which are on the bottom. So those flame retardants are used in a variety of other flame retardant mixtures as well. They're also used in a whole bunch of other applications, particularly the organophosphate flame retardants. So that's the mixture we work with. And so here's our prairie voles. And so we expose mom to 3 doses of these flame retardants. And then of course the vehicle control that's exposed to just the vehicle. And then we expose her during gestation, and then we expose her across lactation. We raise the pups up to adulthood, and we test the pups. So we run a whole variety of behavioral tests that I don't have time to go through. I'll show you the data from the partner preference test, which is that attachment one. So this is how that works. You take a male and a female prairie vole and you introduce them. So they're together for 24 hours. They usually mate within that 24 hours, and they'll form a pair bond. And then the next day, we put them in this 3-chamber arena where we put the partner in little prairie vole jail, and we put like a sexy stranger in another little prairie vole jail, and then we let the other prairie vole choose, right? So we may have a female choosing between 2 males or a male choosing between 2 females, and we count basically how much time they spend with their partner versus the stranger versus alone. Easy test. So this is what we call a partner preference index. So the more those bars are shifted to the right, the more they prefer their partner. The more it's shifted to the left, the more they prefer their stranger. And so what we see is that the females are perfectly fine with partner preference. If anything, they may show a stronger partner preference. And in the males, regardless of dose, the partner preference is gone. And so if you break it down over the course of the test and look what they're doing sort of minute by minute across the test, I want to focus on the males, which are blue. And so you can see a normal male partner preference is increasing over the course of the test. So they spend more time with their partner across the test. And in our exposed males, they don't show any partner preference at all. This was a 10-minute test, and we got a little bit of wonky behavior in our control females. So we wanted to go ahead and do it again. We've since done it 3 times. This is data from the second time that we ran it. We ran it a third time, so we went a little bit longer this time, and we see exactly the same thing. So here's our females. Females are on the left. Controls are all in black, so they show totally normal partner preference. Firemaster 550 is in gray, doesn't totally normal partner preference. They look exactly the same as controls. The males in black are controls, so you can see they have a stronger partner preference over time, which is what they should do, and not our Firemaster animals. So maybe they show a partner preference at an hour, but the rest of the time they're hanging out with a stranger, they're not with their partner, they're exploring all around the arena. And so we've lost that partner preference, but only in our males, which is, we would expect a sex difference, so that's not terribly surprising. So now we can go back to the research domain criteria and try to look at mechanism and try to understand what's broken and why, right? And so here's your list of molecules, cells, and circuits, but we can do better than that. We can make a circuit map, and we can try to understand which systems we're interested in and where the nexus points are within those systems. And so we're focusing on a bunch of different endpoints in the lab. I'm just going to show you some data from the nucleus accumbens. So nucleus accumbens is where the dopaminergic neurons are projecting to from the ventral tegmental area. That's where you literally get addicted to your partner, is your ventral tegmental area, is their nucleus accumbens. And so we started there. We took a couple approaches for this. This was a master's project that was done by Mason Nelson in my lab to look at gene expression in the nucleus accumbens. This is females, and so you can see the volcano plot that's there. And what we noticed right away is that one of the dopamine receptors, DRD1, was downregulated. That's interesting because DRD1 inhibits partner preference. So remember, in females, it might actually be stronger. So the fact that the dopamine receptor that might inhibit partner preference is down makes sense, right? But we also see changes in 2 serotonin receptors in our females. And what I didn't tell you is that Firemaster 550 makes our females incredibly anxious. And so they might be anxious, or they might be showing strong partner preference because of that anxiety. So they're getting social buffering. If you're freaked out, right, you want to hang out with a buddy. Like if you go to physics class and you're scared out of your mind and you're like, this test is going to be awful, you're going to sit next to your buddy. That's social buffering. So we think that's what our females are doing and why their partner preference is a little bit stronger. Now we're going to look at males, and I think you noticed, I hope you noticed when I switched the slide, the volcano plot looks really different, which again emphasizes that male and female brains are quite different. We don't see any changes with the dopamine receptor in the males, but we do see similar changes in serotonin receptors with them as well that we're trying to understand. And then we also see changes in 2 genes that we were not expecting. So one is an adrenergic beta 1 receptor, which we were like, what? That's important for heart. But it's also making us think about neuroadrenergic systems in brain, which had never come up as being a mechanism for how you could get changes in partner preference. So we're sort of thinking about that and working with some folks at NIH to try to understand that. The other gene that's interesting to us is palmitoyltransferase, which is required to put estrogen receptors on the membrane. And in this brain region, all the estrogen receptors are on the membrane, and they're there in both sexes. So it's possible that estrogen receptors may be important in the male for this behavior. And that's not terribly surprising. Even though we think of testosterone as the male hormone and estrogen as the female hormone, we all have both, and they all do important things in both of us. And so it could be that in this case, the estrogen receptors are playing a role in the males. And so we're also focusing right now on quantifying both oxytocin receptors and vasopressin receptors, not just in this brain region, but in other brain regions because of that dimorphism that I showed you, that's highlighted there in red that I showed you before. We're in the slow part, which is slicing all the brains in order to get the tissue that we need to do the autoradiography, but we're hoping to run it this summer. And then I have a student in the lab that's working on social recognition, because if you don't recognize your partner, if you don't remember that's your partner, you're not going to hang out with them. So we're looking in hippocampus, and because we're focused on males, we're also looking at lateral septum to see if we have differences there that might explain the loss of this behavior in our males. And so hopefully what I've hopefully I've convinced you is that if you use the research domain criteria, you can step away from thinking about disorders, which is really overwhelming, and instead focusing on all of these traits, right? And so rats and mice and zebrafish and other types of species might be great for looking at behavioral traits that are sensoromotor, for example, like fish swim. So you can look at motor in fish, right? Positive, negative valence, things like anxiety or fear. But for social processes, we've tried to fill a void by using this really unique species so that we can look at behaviors that are not displayed in some of these other animals. So there's just some other species that we all love to work with. We do work with rats. We do work with zebrafish. We don't work with frogs. Another species that's interesting is Peromyscus. So there are Peromyscus mice that are both monogamous and non-monogamous. So other labs are using Peromyscus as well. And then there's the diversity outbred mouse. So you can take these mice that have this massive genetic diversity and then expose them to a chemical and see when you get a trait to look at G by E. And so I would just encourage all of you to think about the tool that you need to address the question of your interest. And maybe it's not the standard C57 Black 6 mouse that's in like everybody's vivarium. And then the goal within my division at NIEHS is to we want to screen chemicals, 350,000 chemicals. I can't do them all in a prairie vole, but if I know what's broken, then maybe we can do high throughput, that circuit on a chip, social circuit on a chip type thing, and try to narrow down what are the chemicals that are putting developing brains at risk. So this is sort of one of the long-term goals that my division has. So we're translating our animal work into platforms that require fewer or no animals at all. And then I alluded to this at the beginning of the talk, but there's lots of ways to injure the developing brain, right? So if mom gets exposed to a certain type of chemical mixture, yeah, we typically think it gets through the placenta, hits the brain, developing brain has a problem. But that's not always what happens. It may be a change in placental function, and it may be a change in mom's health. And so while we're thinking about how to develop these non-animal platforms, we have to think about that. We have to think about how to model these whole systems when we're trying to figure out which chemicals may be harming the developing brain. And so I encourage you to take a look at the RDoc. I think it's a powerful mechanistic framework for establishing causality for these traits. And then understanding those mechanisms will enhance these non-animal model development. So we're focusing really heavy right now on oxytocin receptor and vasopressin receptor. I hope I've convinced you in this really short time that Firemaster 550 sex specifically impairs social emotional behaviors. Please always include sex as a biological variable. It's really important, and you may see different things in each one. We see that loss of attachment in males, but not females. And in our females, we get really high anxiety. So we get different types of traits. And we expect that because the underlying circuitry is sexually dimorphic. And please pick an experimental model that best addresses the question of interest. If you want to use a funky, fun prairie vole model, like there are many of us in the world that will collaborate with you, so don't be afraid. So Peromyscus is another useful species. People working on zebrafish. So don't be afraid to reach out and collaborate. That's how all of us build a good science program, right? And the RDoc can aid you in your choice of model organism, right? So you can start there and say, what do I need? And then build backwards. And so with that, I'm going to acknowledge the people that do the work. So I'm at NIEHS now. I've been there for a little over 2 years, but my animals are still at NC State, which is 20 minutes down the road. And I collaborate pretty heavily with the folks at NC State. And so I have to give big thanks to John Meitzen, who's sort of the keeper of the colony, Heather Stapleton, who is the environmental chemist that really helps us determine which chemicals to work on, prepares all our dosing solutions, and makes sure we're doing human relevant traits. And then AJ Newell and Jinyan Cao were with me for a long time at NC State and moved over to NIEHS, and they've just been a tremendous pleasure to work with. And so with that, I think I've kept to 30 minutes so that we can have time for questions. And so I hope that was useful and helpful, and thank you very much for your time.
Thank you, Heather. I guess I'll start us off with a question that I have. With the prairie vole model, have you witnessed any sporadic development of autism-like traits, changes in affective behavior that is not linked to any kind of experimental protocol, but just sporadically occurs within a colony?
So really we've only been heavily focusing on the attachment and affiliation traits, and they vary just like humans do. So spontaneously, I think you saw a little bit in those plots that when we do partner preference, some will spend their whole entire time in the test with the partner and others maybe 60, 70% of the time. So we get a little bit of that variability, but we haven't really looked at we don't see any obvious like stereotypy or those types of things in our animals. If we do, we tend to not use them for the tox experiments because we know something else is up with them. So I don't know if that answers your question.
Yeah, it does. What I'm really thinking about is I remember being presented with all of these different animal models of autism and thinking, at most, this is going to be representative for maybe 2% of people who have an autism diagnosis. So, how can we get at modeling everyone else without needing to be exposing them to a chemical milieu? Is there any other cause that we know of that could help that to arise in a colony? Um,
Yeah, I mean, I think there's a lot of things that contribute to these behavioral changes besides just chemical exposures, right? And the autism umbrella is so big that, like, even that oxytocin nasal therapy only works for a small percentage of kids. Um, so, for example, I forget the exact number now without looking at my notes, but something like 20 to 25 to 30% of autistic individuals have higher blood levels of serotonin. So your gut makes way more serotonin than your brain does. That's going to be a different endotype than a kid who doesn't have that. Um, and I think we probably see that variability in the animals as well. So there's going to be a lot of different ways to get to a disorder, which is why I like focusing on the behavioral traits instead. Like if you have impaired social memory, it doesn't matter what neurodevelopmental disorder you call that, right? It's impaired social memory. So if we figure out a chemical that impairs our social memory, I'm going to worry about that chemical, right?
That was a great talk. Um, it, I'm wondering specifically with the autism spectrum disorders that you're studying, the role of the placenta, because I've seen you talk about this before with a variety of, um, of adverse behavioral outcomes, but specifically with autism, what have you found on with the role of the placenta?
Yeah, so those organophosphate flame retardants we have found probably do not get to fetal brain at the level that humans are exposed to. And we see them so these are all animal model studies, because we can't look at a human fetal brain. Obviously, that would not be ethical, right? So we've exposed at really, really high levels, and we don't see the chemical in the fetal brain. But we see it at higher levels in the placenta than we see in mom's blood, which means that the placenta is either storing it or it's deliberately pulling it out or it's keeping it from entering the fetal circulation. And then the question is, what does that chemical do to the placenta? And what we've seen it do is disrupt the placenta's capacity to make serotonin. So for most of pregnancy, the brain is fully dependent on the placenta for all serotonin. It's one of the circuits that develops absolutely last. So if you disrupt the ability for the placenta to develop serotonin and pass it to the fetus, you're gonna have problems in that developing brain. And we have seen that. We think it's also disrupting the production and transport of other neurotransmitters like dopamine and norepinephrine. We're repeating those experiments right now. And in that case, we do rat because we know more about rat placental physiology than vole. So need a different animal model to answer the question. Um, and try to understand how these different chemicals are impacting the placenta. If it's lactational transfer, we do see these chemicals in the offspring brain. So we know that lactational transfer can go direct to the offspring brain, but placenta, at least for the organophosphate, maybe not.
So I have another question. Have you looked at changes in the expression of specific transporters, like PGPs, OCTs, OATPs, anything like that in the placenta?
We have. So we've taken a couple of different approaches. We've taken transcriptomic approaches in the brain itself, and we've taken transcriptomics approaches in placenta. We don't have any obvious, you know, something that's upregulated or changed in the transporter area as to what it would be doing with these organophosphates. We're starting to wonder if maybe it's metabolizing things differently. And maybe so the male and female placentas have different levels. And it doesn't matter uterine position. So it looks like the male placenta attached to a male and a placenta attached to a female may be handling it differently. So part of what we're trying to do by repeating these high dose experiments is to try to get at that a little bit more as to what is going on. And we'd really like to know if it sits in extracellular vesicles. So we're trying to figure out if there's a way to do that. We tried to use MALDESI, which is a form of mass spec where you take a whole slice and you put it on a slide and you kind of sample that whole slide and see if you can see the chemical. And I think it's just so low, we really couldn't catch it. So, I mean, if anybody is interested in that, we would love to know a little bit more, but we're trying to figure out if maybe it's in the extracellular vesicles, but we're not sure.
It's an interesting project.
There's always more to do.
Yeah, it seems very difficult. We have a comment in the chat from Melissa. She said, not a question, but a comment. The way in which you apply the RDoC sounds like a sort of bottom-up system of developing a model which allows for a more direct and well-mapped out experimental process. This is a process that could very well be applied across many topics in biomedical research. Very cool and impressive.
Oh, thank you, Melissa. Yeah, I think so too. I mean, so we have these standards now for reporting animal data. Like we use the ARRIVE guidelines, which is almost like a checklist of what to put in your methods to make it reproducible. But I like the RDoC for the way that you just characterized it. It's a really good framework to design your experiment from the bottom up. And it's highly translatable because it's been crafted with human data. So thanks. I appreciate that.
I think a challenge that maybe half the people who are here experience, because a lot of us are in the same research center, is how to model headache and pain well with rodents. And I've certainly had a problem where my animals will show significant effects when it comes to a photophobia assay, but on things where it's a human rating their behavior, there's so much more buffer there that the signal just gets lost and we're not seeing significant changes.
Oh, yeah. Yeah. I know pain is in the RDoC, but we haven't done anything with pain. So I haven't dove into those circuits to see if there's anything there that's useful. But I know pain, you can't even tell when your pet is in pain. It's so hard. Not always, anyway, not easily.
Yeah, it's definitely challenging, but also regulated via the PVN, so could be crosstalk.
Indeed, it is.
Okay.
And the amygdala, always the amygdala.
Does anyone else have any questions for Heather?
Hi, I have a question. Sorry, I'm joining so late. I really wish I could have attended, but I got pulled away to a retirement party for Dr. Catherine Smith. Yeah, so I'm really sorry I didn't get to see your talk, but I am interested in the topic of autism. Really, the only thing I know about it is a hypothesis that a TREM3 protein. One of the hypotheses is that the TREM3 protein is like cutting the mushroom, like those little mushroom bodies along a neuron. And I was just wondering, in some of your analysis, are you looking at some of the proteins in the fetuses?
Oh, that's a good question. So, I mean, I think one piece is there's probably multiple ways to get to autism, right? And a lot of genetic variants that are associated with it. So there's probably a bunch of combination between a genetic risk factor and a chemical exposure that's going to get you to the disorder. We don't necessarily do that G by E kind of work, at least not yet. We haven't looked at, I think what you're talking about is spine density on some of these dendrites. We haven't,
Yes.
honestly, we haven't looked at spine density yet, but we've talked about it. So John Meitzen, who's our collaborator at NC State, is doing some electrophys work. So I showed some transcriptomics data from nucleus accumbens, but he's also doing electrophys in a nucleus accumbens. And so that's the region of the brain where the VTA, the dopaminergic projections are coming in. And it should be very sensitive to oxytocin and vasopressin and respond accordingly. And what he's seeing in electrophys is it does not. And so the issue is going to be why. And he's going to look at some of those medium spiny neurons. And then a lot of the hippocampal work that we're doing, we're going to focus on CA2 because that's where the oxytocin receptors sit that are associated with social memory. And there are a lot of chemicals that seem to just wipe out those dendritic spines or it makes them less flexible because that spine density is changing all the time. The number that you have, how fat they are, like the characteristics of those spines are constantly changing. They should do that. And it looks like some chemical exposures mitigate, like eliminate that plasticity. So we've been talking about how to do that in hippocampus, but we haven't done it yet. So that's a great question.
That's really cool. Thank you so much.
Yeah, you're welcome.
I think that actually connects to Caitlin and I took this class together when we were first starting the graduate program, where autism was covered as potentially too much synaptic pruning or too little. And that was the mechanism that was explained to us. And then we just moved on. And all the students were like, what? What are you talking about? That makes no sense.
Well, it's common in some autistic kids. They call it brain overgrowth, right? The brain's bigger, it weighs more. And so I think there are some where the pruning hasn't happened. There's also a big switch that needs to occur in your brain where you need to dump a bunch of gray matter and replace it with white matter. That's basically what you're doing as a teenager is substituting gray matter for white matter, and you're refining that over the course of your life. And schizophrenic patients have the same issue, right? And so there's a lot of mechanisms there. I think the key thing to remember for brain is nothing is linear. Everything is a curve. So having too much is a problem. Having too little is a problem. The way that I like to characterize it to toxicologists, because they love linear things. So anything endocrine and they're like, no. If you think about thyroid hormone, right? Someone's hyperthyroid has very different symptoms than someone that has hypothyroid. You want that optimal sweet spot, right? So it doesn't surprise me that some autistic individuals will have too much pruning and others will have too little pruning. Pruning seems to factor in schizophrenia too, and it's also important in that switch from being largely excitatory to inhibitory neurons. That's why it shows up in adolescence. So there's just a lot here for us to probe into, and I'm hoping some folks are interested in neuroimmunity because that's become a really hot area because the immune system, like microglia, does a lot of this pruning and does a lot of this responsivity. And so that may be something that's also gone awry with some of these chemical exposures is that interaction between the immune system and brain.
Have there been changes in estrogen receptor expression in those microglia?
In the microglia, I don't know. In the microglia, we just finished doing an estrogen receptor expression study in the voles that we just published, thinking we'd get big changes in males. It's all really subtle, but it's in that lateral septum, which is it's what we predicted. And we're going to try to repeat that in the rat, but we haven't looked. We know we have differences in the number of microglia. We only see it in males and we only see it in the prefrontal cortex. But we didn't look at estrogen receptors, so that's an interesting question. It's probably something we should do.
Heather, what other neurological disorders are the voles good models for?
So they're also used for grief, and because if you separate them, they'll show signs of grieving. But they're mostly just used for the social stuff right now. So it's wide open if you want to use them for other things. Paternal care, it's used a lot for paternal care and trying to understand that circuit for paternal care and sibling care. So a lot with empathy.
So with your overview slide I've seen before where you have, I think you had at the beginning of this talk with many different mental health problems that are overlapping. Were there any other ones besides autism for which the kind of social cohesion part is really important and therefore the prairie vole would be a good model for that?
So some of the conduct disorders and personality disorders have they either have a negative affect, which is part of it, or they have complete social detachment. And so the fact that our males are not showing partner preference at all is actually worse deficit than we thought and would overlap with some of those other disorders. We tried a double hit experiment. So in prairie voles, you need mom and dad to raise the offspring. If you take dad away, and this was experiments we didn't do, Larry Young and others did these experiments. If you take dad away after birth, the offspring have social deficits. So if they're raised by a single mom, single vol mom, they have social deficits later in life. So we combined taking dad away with exposure to chemicals, thinking we would make the situation in males worse and maybe unmask some challenges in the females. And we got almost nothing, which is really interesting, which means we're either already at this ceiling with our males, and that's as bad as you can get. And females just seem resilient. So that was our attempt at a two-hit. We didn't get anything.
One thing that I think might be really cool to look at is even if there's not a really huge change in estrogen receptor expression in a region, I wonder if you would observe a change in the Jupiter coupling potentially. It is a bit more promiscuous as far as GPCRs go.
Yeah.
So I would wonder about that.
Yeah. So the other thing we're doing now too is we're starting to dabble in spatial transcriptomics because it's hard to go one, region at a time and try to look at things as more of a gestalt and more of like gene families. And there was a paper that came out in Science this week. It's a human-based study where they looked at sex differences in cortical neurons, and there's way more than anybody even thought. And what we've noticed for years, and it's reiterated in this paper, is they're small. So these meaningful differences between males and females that distinguish males from females in brain are quite small. So if you do an RNA-seq experiment and you correct for multiple comparisons and you do all the things you're supposed to do and all of that, you get very few differentially expressed genes. Well, yeah, if a 20% difference distinguishes a male from a female, you're never going to see that in a regular RNA-seq approach. So we've started using these machine learning approaches and asked the question of the model, what distinguishes exposure from non-exposure? Can you tell us what all the exposed animals have that none of the unexposed animals have? And we're getting these smaller gene, like low expressing genes that we don't catch in normal RNA-seq. So I would encourage you to try an alternative approach. One that shows up is a lot of genes involved in mitochondrial function. So we're wondering if it's an energy dynamics issue. And of course, a lot of these neurodevelopmental disorders, people are starting to realize that there's mitochondrial dysfunction going on. And so that is intriguing to us. And it's another reason we've started to move to hippocampus, because it's going to be extremely sensitive to that, if that's really what's going on with these guys. So we'll see. We weren't expecting that. That's what we got.
That's really cool. I feel like you could build networks on that. You couple that with behaviors, and you can even like factor in things like how long were the prairie voles mating, like how long were they introduced, how old is the mother, how old is the father, what are the conditions, are they happy, are they not happy? That's so cool. That's so exciting.
I know. I would love to put them in little semi-natural environments too, so that they're all nice and in their little world. But
Yeah.
Space.
That would be so cool.
Yeah, because, we've known for a long time that advanced parental age is a risk factor for autism, right? But maybe that's not just age. Maybe there's something about getting older, right? So like there was a talk at NIH this week on the autoimmune antibodies. So moms that have these autoimmune antibodies, their offspring are at higher risk. That maybe is part of that big golf umbrella that gives you autism, right? That's going to be a totally different thing potentially than what we see. We don't see any of the autoimmune stuff in our animals. So that's going to be a totally different mechanism, right? But I think you're right. I think it's cool to play around with these different things and start to make these maps as to what's potentially going on.
I have another question. This is a little bit in a different direction, but I've heard so much about the gut microbiome and autism and how there's this amazing connection and how it's bullshit and back and forth. And mechanistically, what do we actually know about the connection there? Is it that a disrupted gut microbiome is more likely if you are autistic, or is there some connection demonstrating potential causality?
So I think the chicken and egg problem is tough. I don't know that anybody's really solved it yet. I totally believe it though. There's, at least for some, right? We have to think about autism as having multiple endotypes. I think that's probably one. So there is a group of patients that's been known for a while that they basically have a leaky gut and they don't break down foods necessarily the same way. And part of them, their gut makes a lot of opiates. And so they end up craving white bread and carbohydrates and a lot of foods that digested improperly with your gut microbiome make opiates. And so they're basically on opiates, right? That's a small subset of individuals, but it's very real. There's now a lot of talk about Parkinson's also starting in the gut, and that a lot of biomarkers now predicting Parkinson's and some other disorders are showing up in the gut. And so I find it a fascinating area of research. I mean, the vagus nerve has been an obvious connection for a really long time. But there's now other direct connections between the gut and the brain that people are really exploring. We personally don't do it, but I find it really cool. And it wouldn't surprise me if we see a more connection between the two. But I don't think it's going to explain all of it, right? It's going to be one endotype or a couple of endotypes. Well, we're in the pause, right? Like, sometimes when I give this talk and it's longer, I show the work of Renee Spitz, who was the Austrian psychologist in the 1940s that observed babies that were born in a women's hospital versus babies that were born in a women's prison and asked which ones came out better. And it was actually the kids in the prison because they're on the floor and they're playing with other kids and they're, whereas in the hospital, they're all swaddled and they're all like individual in their little box. And that's basically like psychological torture for newborns. And he was the first one to really show this. And the kids that grew up and developed problems later, the common comorbidity was immune. And so that goes with that connection between the brain and the gut. And in the 40s, right, like nobody had the capacity to look at, the microbiome or anything, but now we do. And so it doesn't surprise me that those things all go together and that there would be some comorbidities there.
Yeah, that's really interesting. Something I've also been thinking about lately is more on the neurodevelopment side, that we have these key windows where you're really susceptible, and there's neurodevelopment as, in utero or on a few postnatal days for rodents, but then also the transition in puberty. And there is an increase in neuroplasticity and pain disorders and quantification of pain. And so one of the things that I'm curious about is if the quantification of pain changes based off of your exposures, if you have a different ability to regulate that over time.
Interesting. Yeah.
Yeah.
Well, I know that the pain pathways and sensitivities also change with age. And so aged individuals have different pain perception than non-aged individuals, which I care about more the older I get.
Yeah, I think there could be a lot of interplay there with like the immune axis and then neuroplasticity and exposures.
It's definitely a big area right now.
Yeah.
Well, if anyone on the call is interested in more about prairie voles, we have them. We give tissues away. We collaborate with people all the time, particularly if you want anything from the neck down. So we don't work on heart. We don't work on orexin neurons or any of that kind of stuff. So we're always happy to collaborate and share with people. So I'm just putting that plug out there. If anyone is interested, we ship pilot tissue everywhere.
Awesome. Do we have any more questions before we close up the call? All right. Well, thank you, Heather. This was really interesting, and I'm glad that we were able to make it happen. And a pretty exciting tool that I think all of us can use.
Well, thank you. It was a pleasure to introduce it. Thank you also for giving me the second chance to present. And it was great having an opportunity to talk to everyone on the call. Really appreciate it.
Awesome. All right. Bye.